Queen Nerdling here, coming to you today to talk about signal transduction pathways. So let's get started. So first of all, I want to discuss the two different types of receptors that you might encounter in your studies of cellular communication. So we have intracellular receptors, and just like they sound, those are receptors that receive signals inside the cell, meaning that the signaling molecule, or the ligand, actually goes through the plasma membrane and then eventually will dock with its intracellular receptor. And then we have plasma membrane receptors, like these guys right here. This is when a ligand has to dock with an external receptor, and that ligand can't actually go through the plasma membrane itself into the cytoplasm or docking with that receiving protein inside the cell. So again, we have intracellular receptors, such as these, where that ligand can actually go through the plasma membrane and then dock with an intracellular receptor. And the other class, we have our plasma membrane receptors, where ligands will actually dock with the receiving protein, and then the message gets relayed, and the ligand never actually enters the cell itself. So the chemical properties of the ligand, or the signaling molecule, actually determine what kind of receptor it's going to bind with. So first, we're going to discuss our intracellular receptors. Now, these are associated with hormones or lipid-soluble ligands, so fats or our lipids. Now, these are typically non-polar molecules, and because they are not charged, they easily go through that plasma membrane. So that hormone or lipid-soluble ligand passes through the plasma membrane, and then it will bind with an intracellular recepting complex. Now, this ligand receptor complex is then transported by motor proteins on the cell's cytoskeleton to the nucleus. So we have to get that signal to the nucleus so that that signal is actually expressed. And nothing is going to happen unless that signal is expressed, and it has to be expressed by DNA receiving it, and then the DNA will make a complementary messenger RNA strand, and then the messenger RNA will take that message that the ligand sent, and a response will eventually occur. So the ligand receptor complex will enter the nucleus. It binds to DNA on promoter sequences, causing those genes to be transcribed into that messenger RNA strand. The messenger RNA strand is then going to be able to leave or exit the nucleus, exons, flashback to gene to protein, and this is called a transcription factor. That messenger RNA leaves the nucleus to be translated on ribosomes into a protein. So then those proteins can go out into the cell and perform all of the numerous types of functions that they're meant to do. So now we're going to talk about plasma membrane bound receptors. So this is a receptor that's actually embedded into the membrane itself. The ligand binds with the receiving protein extracellularly. So again, the ligand does not actually go into the cell itself. It has to bind to these receiving proteins. The receiving protein changes shape, and it transmits the signal inside. So when I say change shape, the fancy schmancy biological term for that is it undergoes a conformational change. So it changes in shape, or the conformation changes. And it's very similar to our enzyme substrate complex. So just like an enzyme binds with its substrate and undergoes a slight conformational change, the same situation occurs here. If we're pretending this is my ligand or signaling molecule, and this is my little end of the receiving protein, binds with my signaling molecule, and then a conformational change will occur, and that will create this transduction of that signal. So as far as that occurs, the second messenger is then activated by that receptor protein changing shape, or that conformational change. Now the signal transduction cascade will now begin. So this is basically a bunch of proteins that get phosphorylated, meaning they gain energy, or they're getting energy, and those get energy all in a row. And so we get all these molecules that have been phosphorylated, and then we have secondary messengers that are proteins in that transduction or pathway, and it helps to get this cascade going. And so this moves the signal from the cell membrane to the nucleus through this signal transduction pathway or through the transduction portion. So we have that signaling molecule, it binds with our receiving protein, it causes a conformational change. Now that conformational change will then activate a secondary messenger. That secondary messenger will then perform a function, which might be creating cyclic AMP. And then that cyclic AMP might go and activate a protein kinase, which then in turn activates another protein kinase. And we have all of these being activated at the same time. So that initial signal gets amplified and it gets bigger and bigger and bigger and bigger until eventually that signal gets sent to the nucleus. And that is where we can actually have a cellular response when it hits the DNA, and the DNA can actually send that message to be upregulated or transcribed and then translated into a fully functional protein. So there are secondary messengers. One of the most common ones that you'll see in AP biology and probably read about in your textbook is called cyclic AMP. And you might see the abbreviation of CAMP. Now, these are molecules that help to relay and amplify the intracellular signal. So there are many different types of secondary messengers. You do not need to know all of them, but you might just want to know an example of one, and you need to understand their function so you could identify them, say, if you were given a crazy free response test. So giving you a little bit of an example of epinephrine-stimulated CAMP or cyclic AMP synthesis. So we have our signaling molecule. This is called if you said ligand, you're correct. So specifically, this would be epinephrine. Now this is going to dock with our receiving protein. Once it docks with this receiving protein right here, it's going to cause a conformational change. Now the conformational change basically gets relayed, and if you see, these guys right here are basically together. Once that signaling molecule or ligand docks with the receiving protein, it causes a conformational change. So in this case, this conformational change releases what we call the alpha unit of this G protein. So this alpha unit will then actually activate this guy right here. So this is an inactive adenylyl cyclase. Now that suffix ASE or ACE should kind of give you the indication that this is an enzyme and it's going to do something. So it activates adenylyl cyclase. Now adenylyl cyclase then goes on to create cyclic AMP. So cyclic AMP then goes on to initiate interactions with protein kinases, and eventually those protein kinases will activate a hydrolytic enzyme. That hydrolytic enzyme will then basically go into our glycogen stores and start breaking those glycogen up into glucose molecules that we can then utilize in energy, and now we can run and we can go into our fight mode or our flight mode. So now we're going to talk a little bit about that phosphorylation cascade that I just discussed. So the transduction pathways are responsible for amplifying messages. These essentially mean that the subsequent phosphorylation, meaning adding phosphates, so phosphorylation, we're adding phosphates, of the proteins moves that signal from the membrane to the nucleus. This increase in the strength of the signal ensures that that message gets to the nucleus so that the DNA can play its role in telling messenger RNA to get the message out, make this protein so it can do this function. And this is also an example of a positive feedback. So again, we have our signaling molecule, docks with our receiving protein. This receiving protein gets activated. Now this might activate a secondary messenger, such as cyclic AMP. So it might activate the enzyme adenylyl cyclase. Adenylyl cyclase will then start creating our cyclic AMP, which is our secondary messenger. That cyclic AMP then might activate a protein kinase, which then activates another protein kinase. And if you notice, each time one of these is activated, we're utilizing that energy from a high energy phosphate bond in ATP. So that's how we're getting that transfer of energy. Eventually, this cascade is going to elicit a cellular response. So the final protein activated in this transduction pathway is going to enter the nucleus. This is going to bind to the DNA promoter sequences, causing genes to be transcribed into messenger RNA. Messenger RNA is then going to leave or exit the nucleus, and it is going to find itself a nice little ribosome so it can get its protein synthesized, and then that protein will go out into the cell and perform whatever function it's supposed to perform. And all of that occurred because of that signaling molecule saying, we need to do blank, and a response will eventually occur. Now, there are 3 examples of plasma membrane receptors that I want to discuss with you. So the most common by far is the G protein coupled receptor. So G protein coupled receptors bind to the G protein, or I should say the ligand binds to the G protein coupled receptor. So that G protein coupled receptor changes shape and it phosphorylates GDP on that G protein. So the phosphorylated G protein moves toward an inactive secondary messenger enzyme in the pathway, and then the phosphate from that GTP is transferred into an inactive enzyme, thus activating it, and the activated enzyme can start the transduction pathway. So showing you what I just said on the diagram. So we have right here our signaling molecule. It docks with this G protein coupled receptor. Now when it does this, it activates it, and a little component of that G protein actually moves. So because it got phosphorylated, meaning it gave some energy, now it can actually move. So this would be an inactive enzyme. It's there, but it's not ready to perform its job yet. So once this is activated, because it bound with that ligand, this little alpha subunit is what we call it, it can then activate this enzyme. So it could activate an enzyme called adenylyl cyclase. Once adenylyl cyclase is activated, it can then start pumping out AMP, or cyclic AMP. Cyclic because of the structure, literally cycle, so cyclic. Cyclic AMP, which can then go on to participate in that phosphorylation cascade where it phosphorylates a protein kinase, and another one, and another one, and another one, until we eventually get a response. Now, as soon as this ligand detaches from that G protein, this little guy right here would go back into the inactive state, which would create this, which would allow that enzyme to go back into its inactive state, so it would stop producing cyclic AMP, which would stop phosphorylating protein kinases, and that response would no longer be needed. So that kind of helps to regulate that signal transduction pathway. So like I just said, once that G protein is phosphorylated and that secondary messenger enzyme does its role, that G protein will go back into its original inactive state or conformation until another ligand binds with it. So the second type of receptor I want to discuss are called tyrosine kinase receptors. Now, tyrosine kinase receptors are dimers, meaning they have 2 of these subcomponents or subunits. They have 2 proteins that associate together to form a quaternary or fully functional protein complex. So once the ligand binds to that tyrosine kinase receptor, it causes it to dimertize, aka bind together to form that quaternary structure, or the fully functional version of that protein. So the dimerized tyrosine kinase receptors phosphorylate each other. So they basically activate each other. And then that phosphate is transferred from ATP to the tyrosine amino acids on the tyrosine kinase receptors. And this activates those tyrosine kinase receptors. Now, once those activated receptors are bound, they can interact with secondary messenger proteins and help to initiate those cellular responses and amplify the signal so it can eventually reach the nucleus. So the last type of receiving protein I want to talk about are the ligand-gated ion channels. So these are extremely important for nerve impulses, meaning how we send signals in our nervous system to tell our bodies what to do. So these receptors are actually channel proteins, and the channel proteins are normally closed, meaning that the ions aren't just coming into and out of it as they want. So it's closed off to these ions or charged particles. So when a ligand binds to that ion channel, it actually causes, again, a conformational change, meaning this time it opens that channel. So ligand binds, conformational change occurs, and all of these ions can rush in. So they diffuse into the cytoplasm, and this causes a membrane potential. It actually causes an electrical charge across the membrane, and that's how we generate an action potential and how a message gets sent from one neuron or nerve cell to the next. So kind of shifting gears a little bit, I just want to briefly retouch on cell signaling and evolution. So most living organisms have conserved sets of signaling pathways, and this indicates that the signaling pathway arose in a common ancestor, and it was passed down to the descendant species. And this is evidence for evolution. So whether we're talking about prokaryotic bacterial cells and quorum sensing, or whether we're talking about super advanced multicellular organisms such as dogs, they all use cellular communication through signal transduction pathways. And so as far as interacting with our environments, we have to have a way to interact with our environments and to maintain homeostasis. So all organisms are able to acquire information from their environment. Again, whether we're talking about a bacteria and quorum sensing, or whether we're talking about giant multicellular organisms such as myself. So a stimulus is anything in the environment that is going to elicit a response from an organism. So for example, I live in the Houston, Texas area. If I go outside in the middle of the day in the summer, it might be 100 degrees outside. So I have to have a response to the stimulus of the heat. Now my response is going to be to sweat, because when I sweat, it's going to evaporate and help me cool off, again, regulating my body temperature. But all of that occurred because of signal transduction pathways telling my body what I needed to do to the stimulus that I just encountered. So the next part is the response, or the response from the stimulus. And this is the ability of an organism, so in my case, myself, to adapt to that heat, to respond appropriately to the stimuli that I just incurred, and contribute to my overall fitness, meaning my overall survival. If I lacked the ability to sweat and to help cool myself down, I might die. So another example of how signal transduction pathways are used would be mating in yeast cells. So they release pheromones in the environment that trigger the mating gene expression. So the first thing that occurs is we have signal reception. They receive this signal that they need to mate. So next, they have pheromones from each of those yeasts that bind to the surface receptor on another yeast cell. So a signal is then transduced. So there's this cascade of internal signals that happen in here. And eventually it causes a response. In this case, the response to all of those mating pheromones for those yeast cells to kind of get it on is for their cells to fuse so they can mate. And that is a result of the pheromones being released into the environment, causes a transduction cascade of internal signals, and eventually the response of it's time to mate, and they will find another yeast cell to fuse with and mate. So another example of signal transduction would be the SRY gene on the Y chromosome in males. So expression of that gene, that SRY gene, initiates testicular development of undifferentiated gonads, meaning these are just our sex organs, and they will either become ovaries or they will become testes. But until that occurs, they're basically just our gonads. So when we have an absence of that SRY gene expression, it allows for the default female state to develop. If we have that SRY expression, it initiates those gonads to turn into testicles. Now again, if we don't have that SRY being expressed, then those gonads will develop into the ovaries. And last example are Hox genes in development. So these are kind of cool genes. These are groups of related genes that basically control the structure and orientation of animals. So how we develop, what develops into our mouth, what develops into our anus, what happens to our tail, so everything. So early embryos of all vertebrates, meaning all organisms that have a backbone, such as reptiles, mammals, birds, amphibians, and fish, we all have vertebrates. And at some point in our embryological development, we all pretty much look the same. So all vertebrates have similar characteristics at a very early embryological development. Now, Hox genes are actually responsible for triggering apoptosis or apoptosis, triggered or programmed cell death. So basically getting rid of cells. Now, in humans, we have different Hox genes. The HOX11 gene triggers apoptosis or programmed cell death to absorb that post-anal tail before birth. Another example of a Hox gene that people have that helps to program cell death would be the cells that are in between our fingers or our digits. Now, those undergo apoptosis and allow for separation so I can manipulate all of my fingers. Now, if those don't work correctly and the correct signal does not get sent for the cells in between these digits to undergo apoptosis, we can actually have abnormalities where people have webbing between their hands because those cells never received the signal to die. So I hope that gave you guys a pretty good sense of what a signal transduction pathway is, some examples of them, and how those signal transduction pathways work. For more videos like this, hop on to www.nerdlingscience.com. This is the Queen Nerdling signing off for today.